additional questions. So now Daniel Lieberman will give us Daniel Lieber. Sorry, I extended the last name. So he's from Takara. Takara is probably the oldest company in the whole portfolio, which we are presenting now. It's from 1925, and as I said the last time, it's a very interesting company because not only focused on the biotechnology of today, but also of the biotechnology of yesterday and tomorrow. They have a very interesting product, a Highland single malt Scotch whiskey, and it's also the largest distiller of traditional social liquor in Japan. So which makes the company very interesting and worth listening to the talk of Daniel. Okay.
Thank you very much, Udo. I guess you can see my screen now. Okay, so thank you very much for the introduction and the time to the opportunity to talk to you today. So today I will focus on our instrumentation for single-cell genomics. So I'm not going to talk about our whiskey and sakes, unfortunately, but hopefully it will be as interesting. So Anas from 10x Genomics has already introduced a little bit the concept of single cell, so thank you for that. So I will not go into the background of single cell genomics. Just kind of a brief introduction about Takara and his involvement in this field. Actually, it goes back a very long time from already in the 1990s when we developed and commercialized the technology which is at the base of all RNA-seq, single-cell RNA-seq protocol, which is the SMART system, which is the template switching tool to synthesize cDNA and then do the amplification of the cDNA. And kind of our involvement in more specifically in the single-cell genomics also is quite old. It's almost 10 years ago that we released the first commercial kit for performing single-cell RNA-seq. And since then, we had kind of thousands of papers that are citing our protocol, more that are even using our technology in general. And we have developed a kind of a wide portfolio of solution for doing single-cell RNA-seq using kind of the SMART-seq, a traditional SMART-seq, doing for stranded analysis, or also for targeting T-cell receptor, for example. What I'm going to talk to you today is the ICELL8 system. This has a kind of more recent history. It was first developed and launched in 2015 by the WebChain Biosystem, which is now part of the Takara Bio family. And in 2018, we developed and launched the second generation of this platform. So the way that we are working is slightly different from other technology in that we use a nanowell chip to isolate the cells and to perform the library preparation. And the ICELL8 chip is shown here in the middle of this slide is, for the people that have never seen it before, it's a 4 centimeter by 4 centimeter alloy chip that has 5,184 wells in each chip. These allow to have a very high throughput, so in terms of well capacity is equivalent to 14,384 well plate. But since we're using limited dilution to make the experiment as quick as possible, not to compromise the cells, we can isolate and process between 1,200 and 1,500 cells in a normal experiment with a kind of theoretical maximum of 1,800. So these allow to have a kind of higher throughput than a conventional plate-based system, but with advantage that we miniaturize the reaction. So we do the reaction in nanoliter volumes, so we use much less reagents. What also makes this technology unique in the space of kind of single-cell kind of complex system automation is that we can perform multi-step dispensing. So we can add multiple reagents. We can perform up to 9 dispensing before we kind of run out of volume, which means that we can perform very complex chemistries within this chip. The ICELL8 is a complete system, so it's made of kind of multiple components. And of course, there is the hardware itself, which is this box shown here, is a kind of desktop device. And there are two major functions that are done within this instrument. So we have a dispenser that we use at different steps of the workflow. So we use it at the beginning to dispense the cells into the chip, and then we use it later to add the reagents and to perform the library preparation within the chip. What we also have in the system is a fluorescent microscope that allow us to identify the location of individual cells and to do characterization of these cells. We have a different software solution that are part of the platform. So we have the software to control the instrument itself, and where we can also use this to develop a specific script so customer can develop their own workflows. We have an image analysis software that allows to automatically identify the location within the chip that include a single cell and to do specific characterization. So we have 3 colors in the instrument, and we can do 2 colors at each experiment. Downstream, we also have a data analysis software. So we have a full pipeline to process the data, and we have visualization software to do data interpretation analysis and also to generate images. And we also have a specific software for our T-cell receptor analysis. As part of the system, we also have multiple chemistries and applications that can be performed on the system that go from RNA analysis, DNA analysis, T-cell receptor, epigenetics, and as I mentioned before, the system is open, so it gives the opportunity to our user to develop and transfer into the system their own chemistries. When we developed the platform, we were kind of trying to cover 3 parts of the experiment. We found kind of 3 issues in the way that single cell genomics experiment were performed. So we wanted to have a solution that will give more control into the experiment, more confidence in the data that were generated in each experiment, and a platform that was flexible and gave flexibility to our users. Today, in the interest of time, I'm not going to go into all the different parts and all the different science behind it. I just kind of want to highlight a couple of points. One is the capability of selecting the cells and only process the wells where we have confirmation that there is a single cell present with specific characteristic that can be done by imaging. Another important feature of the system that is used by many of our customers is that we don't have a limitation in the size of cells that we can capture. This is very important when you have very heterogeneous samples or when you have cells that have very specific characteristics, but particularly they are very large. And also we have multiple applications that are already available, and the platform is open to development for the chemistries. As I mentioned, one of the critical features of the platform is that it allows to have a wide capability to isolate cells of a different variety of different size and different nature. This table is very old, so don't really look at the specifics. We have now many more examples. It's just give you a flavor of what we have been able to process. So we can go from the very small, like nuclei, from a variety of frozen tissues, to something that is completely different, like 3D spheroids that can be dispensed whole into the chip, or adult cardiomyocytes, again, very difficult cells to process due to their size. And we have quite many examples of how the platform can be used to study the heart. And one of the first example was from the Max Planck Institute for Heart and Lung Research in Bad Nauheim, where they first kind of started to use our technology to perform single cardiomyocyte sequencing. And as you can see, the instrument gives the capability of isolating the cells and also to check the morphology and also to identify the nuclearity of these cells, so to be able to really distinguish polynucleated cells from mononucleated cells and to do analysis and differentiate between these. Also, what is very important is that the imaging capability gave the opportunity to quality control the cells that were isolated and really process only the one that maintain the right morphology. And these cells are really unique in their morphology. They have this very characteristic rod shape. And being able to visualize what has been isolated is very important to make sure that there is no misinterpretation in the data. For example, when processing cells that do not have the right shape that could have, for example, gone into apoptosis. Another example of the capability to use kind of phenotypic and morphological information from the imaging was described in this paper from the MD Anderson in Texas, where they were looking at nuclei from breast cancer biopsies. And they used a feature of the tumor nuclei to be larger in size than nuclei from normal cells. So using this feature, they were able to process from a biopsy that only had 41% tumor content based on histopathology, they were able to process for sequencing 99% of the nuclei were from the tumor. And again, this is very important to maximize the sequencing capability and the data analysis only on the cells of interest, rather than having the majority of your samples and the sequencing and the data analysis covered by normal samples, normal cells. In terms of the chemistry, we have kind of a variety of protocols that have been developed and that are available to be used on the platform. Some of them are complete kits that we sell. So we have our kind of SMART-seq full-length RNA-seq chemistry that is available as a full kit. We have also some gene expression kit available, and also a kit for T-cell receptor sequencing. But as I mentioned before, the platform is open, which means that there has been quite a lot of development from also our R&D, but also from our users to develop specific protocols. So internally, we have a protocol for target enrichment or a protocol to combine our library preparation with oxo nanopore sequencing. But there are a couple of examples of chemistry that have been developed by customers like ATAC-seq on cut and tag. And the cut and tag is a very nice example. First of all, it's a very interesting workflow that allows to study histomodification at the single cell level. But as you can see, we are kind of at the second generation. So the NCOF lab is the second generation of this protocol, because they were able to improve the chemistry by adding extra steps to improve the lysis of the nuclei to improve the sensitivity of the assay. And again, this is something that without the technology that do not have kind of open chemistry capability becomes very difficult. And there are kind of other protocols that are in the process of being published. So SMART-seq, as I mentioned, is one of the key chemistry that we have adopted on the system. And there are kind of some specific advantages of the chemistry itself, and also to perform this on the ICELL8. So first of all, the ICELL8 allows to have a kind of high throughput processing, so we can process more cell than conventional.
One more minute, please. And then
sure.
Come to an end. Sorry.
No problem. I'm almost at the end. We can have high throughput. We can miniaturize the workflow. We have a very streamlined workflow, and everything is done in a single instrument, from cell isolation to complete libraries. The other kind of advantages is we have kind of higher sensitivity. So we have a very efficient lysis. So there is a paper also from MDC Berlin that described this, and we have kind of optimized protocol that gives possibility to have very complex library. And again, this is very important when you really want to have a deep characterization of your cells, not only kind of shallow sequencing just to identify the cell types. And again, the full length capability gives information not only about gene expression, but open the possibility to look into isoform, single nucleotide variation, and also fusion of genes. So again, it can give some more comprehensive analysis of each cell that we have we are analyzing. As I mentioned at the beginning, we have a complete suite of software and data analysis pipeline, and I'm at the end. So just to summarize, the ICELL8 is a kind of a all-in-one solution for single-cell genomics that is a kind of a comprehensive and can cover everything from cell isolation all the way to library preparation and data analysis. And that's good science. Thank you very much for your time. And of course, we are at our virtual booth. So if you have any more questions, we'll be very happy to discuss with you further during the lunch, coffee breaks, or tomorrow.
If you don't